Hi everyone, my name is Chang Liu, and I'm a professor at the University of California at Irvine. I'm going to tell you about our work on the development of synthetic genetic systems capable of rapid mutation and continuous evolution in vivo, focusing on the system shown on this title slide. We have made a cell that has what we call an orthogonal replication system, consisting of an orthogonal DNA polymerase that only replicates the orthogonal plasmid and not the host genome. This means that we can engineer the orthogonal DNA polymerase however we want, and its engineered properties will only act on the orthogonal plasmid. In particular, and as you'll see throughout this talk, we can engineer the orthogonal DNA polymerase to be highly error-prone in order to drive the rapid evolution of genes of our choosing encoded on the orthogonal plasmid. To motivate our work on orthogonal replication, let me first ask you to consider the following question. How fast can we mutate a genome? Let's first consider a hypothetical organism with a very small genome, say 200 base pairs. Perhaps this was a replicase at the origin of life. And let's assume that half of those 200 base pairs are essential. At a mutation rate of 10 to the minus 3, what is the chance that an offspring of this organism is viable? Well, to be viable, the organism has to copy its 100 essential base pairs correctly. The accuracy of copying one base is 999 over 1,000, which is 1 minus the error rate. So doing that 100 times means that we have to take 999 over 1,000 to the 100th power, giving us 0.905. So that's a reasonable chance of producing a viable offspring. But now let's consider a different organism with a much larger genome. For instance, a bacterium that has 1 million essential bases. If the error rate was still 10 to the minus 3, then the chance of an offspring being viable is now 999 out of 1,000 to the millionth power, which turns out to be, of course, 0. So, of course, bacteria have much lower error rates than 10 to the minus 3. Suppose instead we made the error rate more realistic, 10 to the minus 7. Then what is the chance an offspring is viable? Well, now it gets back to the acceptable 0.905 chance, which is not too bad. I use this example to illustrate what is a very fundamental inverse relationship between how fast you can mutate a piece of information while holding on to function and its size. For biology, this means that modern genomes cannot withstand high per-base mutation rates because they are simply too large and complex, a conclusion that has been addressed with great nuance in some of the research areas listed here. Now I want to extend this conclusion one step further and argue that because large genomes can't withstand high mutation rates, the focus of evolution has broadened over the history of life. Let me unpack that for you. Here's a plot of the simple analysis I did before on the mutation rates tolerated if we were to replicate either 100 essential bases for the replicase or 1 million essential bases for the bacterium. You'll notice that above a mutation rate of 1 over the essential genome size, you have a low chance of survival. So mutation rates need to stay below this roughly 1 over L threshold, where L is the genome size. Now let's consider what happens to genomes as life evolves and complexity increases. What happens, of course, is that genomes grow in size and get more genes. Because of the 1 over L threshold on mutation rate, as genomes get more genes, this means that the mutation rate per gene must necessarily decrease, assuming gene sizes stay roughly the same. Okay, so now comes the important part of the argument. We know that mutations are the source of evolutionary innovation. So if mutation rates per gene decrease as organisms become more complex over the history of life, this means that the focus of evolution has to broaden from the gene level to the multi-gene or systems level. In other words, modern organisms are innovating and restricted to innovate at the systems level rather than at the gene level. But what if we are interested in innovating at the gene level? For instance, what if you're a protein engineer and what you want to do is rapidly evolve proteins encoded by genes? To sample diversity sufficiently for a protein, you need very high mutation rates per gene, which complex life has locked out by this 1/L threshold. Or suppose you are interested in developmental biology. A very neat feature of evolution is that mutations are not just a source of innovation, but also a record of descent. So you can compare mutations in two different species and figure out how they are related to each other. In principle, you could do the same for cells in an animal and use mutations to map how cells are descended from each other, allowing you to understand development. To do that, however, you again need very high mutation rates so that you can see mutations in the read length of a sequencer. And of course, these high mutation rates are locked out by this 1/L threshold of the host cell's genome. So this brings me to the broader goal of my lab, which is to get these high mutation rates in living cells and break this lock. To do so, what we do is we build targeted mutagenesis systems that direct a high rate of mutation to only particular genes of our choosing, while allowing the rest of the host genome to stay at its necessarily very low mutation rate. We have developed two such targeted mutation systems in my lab, and I will be talking about one of them called orthogonal replication, or OrthoRep, in this video. The idea with OrthoRep is that if I have a second replication system that does not replicate the genome, but is only responsible for replicating our genes of interest on the orthogonal plasmid, I can indeed get to high mutation rates, since 1/L for a gene is much higher than 1/L for the genome. Here's the outline for the rest of my talk. I want to first give you a basic understanding of how we set up OrthoRep. Then I will cover a couple of applications that we've carried out, and then end with some comments on what the future holds for OrthoRep. Let's start with the basis of OrthoRep. To build OrthoRep, we didn't assemble a replication system from scratch, but instead we borrowed from nature. We hijacked a very interesting and unusual replication system found in Clavieromyces lactis. As described in the 80s, some strains of K. lactis have DNA plasmids in their cytoplasms, and it caught our attention that these plasmids actually encode their own DNA polymerases. It was speculated by the researchers that discovered these so-called killer plasmids, because they encode naturally a killer phenotype for the yeast. It was speculated that these killer plasmids were replicated by the DNA polymerases that are encoded on them. For instance, tpDNAP1 is replicating the P1 plasmid. We've subsequently shown that this is indeed the case. Furthermore, these DNA polymerases use an unusual replication mechanism that is not how genomic DNA polymerases work. This mechanistic separation from genomic replication, combined with the spatial separation of the P1-P2 plasmid system from the nuclear DNA, convinced us that the system had the orthogonality we needed to achieve OrthoRep. So we started our effort to develop the P1-P2 replication system. In the first couple years of my lab, we hijacked and refactored elements of the replication system for the yeast Saccharomyces cerevisiae, doing things like encoding the DNA polymerase in the nucleus and figuring out how to encode only our genes of interest on the orthogonal plasmid. And then we did a series of experiments showing that indeed the system was orthogonal, where the special DNA polymerase tpDNAP1 only replicated the P1 plasmid. This gave us our first version of OrthoRep, which allowed us to engineer the DNA polymerase to have a high rate of mutation on the P1 plasmid compared to the genome. However, this higher rate of mutation wasn't quite high enough to tackle protein evolution. So for the next few years of my lab, we mainly focused on getting OrthoRep to run at very high mutation rates through a large engineering effort on the DNA polymerase. This resulted in OrthoRep version 2, which is capable of mutating genes of interest on the orthogonal plasmid at a rate of 10 to the minus 5 substitutions per base, while the genome replicated at its natural mutation rate of 10 to the minus 10. In these years, we also really fleshed out the molecular biology and genetics of OrthoRep, with efforts showing that it was general across yeast strains, understanding the basis for orthogonality, and controlling the strength of expression of genes encoded on the orthogonal plasmid, among other things. I want to show you a slide summarizing the key aspects of OrthoRep with regard to mutation rate. By using different tpDNA P1s, different orthogonal DNA polymerases, we can now choose the mutation rate for the orthogonal plasmid, spanning several orders of magnitude. These are the blue bars. Importantly, these mutation rates do not affect the genomic mutation rate of the host cell, which remains low as indicated by the red dots. Furthermore, the highest mutation rates we've achieved for OrthoRep exceed genomic mutational thresholds, represented as these shaded regions. So we are well into mutation rates that if you tried to get the genome to replicate at, the cell would die. So indeed, much as I motivated earlier, OrthoRep allows us to exceed the low mutation rate thresholds of the cell and should now give us the opportunity to drive the continuous evolution of genes of interest at exceptional speeds. Okay, so let me now show you what we can do with OrthoRep. As you can imagine, we are using OrthoRep to evolve many useful biomolecules. We're also using it to do things like study the rules governing adaptation, uncover and understand pathways of adaptation, and predict evolution in nature. I'll highlight two studies from our recent work in these areas. The very first evolution experiment we did with OrthoRep was to see if we could use it to discover and understand mutational paths leading to drug resistance. We chose to study the malarial version of DHFR, which is a drug target for treating malaria. In particular, malarial DHFR is inhibited by the drug pyrimethamine, which has led to many resistant DHFRs in the field. What we did was we encoded malarial DHFR on OrthoRep in a yeast strain that has its endogenous DHFR deleted. So now if we challenge these yeast cells with pyrimethamine, they will die unless pyrimethamine-resistant DHFRs evolve. Since DHFR is on OrthoRep, the process of evolution occurs very quickly, and in just 13 passages of a 500 microliter culture of these cells, we reach resistance to 3 millimolar pyrimethamine, which is the maximum amount of pyrimethamine soluble in media. We did this in 90 replicate experiments so that we could have some statistical understanding of how DHFR develops resistance. And here's what we saw. This is a table where each row represents a different replicate evolution experiment, and each column represents a mutation observed throughout the experiments. What you can gather from this table is that resistance arises through a diverse set of possible mutations, but at the same time, there are definitely convergent patterns and repeating mutational pathways emerging. For example, almost all populations contain the mutation S108N. At the same time, there are 4 populations, though, that lack S108N. These 4 populations all fix D54N, but there are also many populations that fix both S108N and D54N together, though always with other mutations present. These patterns led us to suspect that S108N and D54N were influencing each other, and those interactions affect the reproducibility of evolution in our experiment. To test our suspicions, we took the five most common mutations observed throughout the experiment and constructed a synthetic fitness landscape defined by those five mutations. In the map here, the resistance for all 32 combinations of these five mutations, including S108N and D54N, are shown, where red means not resistant and blue means highly resistant. Going up the map, we see a general trend that the more mutations we get, the more resistant we are, which is as expected. But the fact that this trend doesn't just monotonically increase across all paths up the map means that you expect adaptation to occur in limited ways, explaining some of our observations in our actual evolution experiments. Here's one observation we could explain. Why did our evolution experiments seem to repeat themselves across many of the replicates? If you look at the first step in the synthetic map, the most resistant single mutant you can choose is S108N. Yet what you find is that in all the high resistance variants on this map, you have the mutation D54N. Interestingly, for most of the mutants where both S108N and D54N are together, you get no resistance, suggesting a conflict between S108N and D54N. The exception is if you have Y57H and C59R also appear, which seems to resolve the conflict between S108N and D54N. So what happens during adaptation is that S108N will likely quickly fix first, sweeping through the population, restricting the paths towards higher resistance to only just 20% of this limited landscape. Then the path is further restricted because the S108N mutant needs to pick up Y57H and C59R before picking up D54N, making the teal path outlined the likely route to resistance. Therefore, our evolution experiments are rather reproducible. But in our evolution experiment, we also observed outcomes that did not have S108N. What was happening there? It turns out that for those cases, what happened was that before S108N could fix, a double mutant, D54N Y57H, arose and took over. This allowed those evolution experiments to follow a different path to high resistance, hitting different mutants on this map that could not be accessed if we first fixed S108N. So our second insight was that the highly resistant outcomes that do not have S108N are indeed accessible, but are usually suppressed through clonal interference by the greedy S108N first mutation. Given these two insights, we predicted and successfully confirmed how to prefer the rare yellow path over the green path. For example, since the yellow path required a double mutation to enter, we could increase population sizes to prefer it, since larger population sizes can sample double mutations in DHFR at the same time single mutations are sampled. Likewise, we predicted that if we recoded S108 so that it takes 2 base changes to turn it into N, we could also prefer the yellow path, which is indeed what we observed. The significance of this study is threefold. First, because we could run so many replicates of the DHFR resistance evolution experiment, we were able to identify rare mutational paths to resistance that would otherwise not have been observed. Second, given the identification of different paths to resistance and the fact that we could prefer one over the other with simple changes, it may be possible to slow resistance by strategically forcing evolution down one path versus another, especially if the distinct nodes on different paths are inhibited by different DHFR inhibitors. Third, and most generalizable, this study shows how OrthoRep could be broadly useful in finding and mapping interesting areas of fitness landscapes through replicate protein evolution experiments that would be difficult to do any other way. Let me now show you another application of OrthoRep, this one aimed at making useful enzymes. In this project, which we are doing in collaboration with Francis Arnold's group, we are interested in evolving an enzyme called s.p.b involved in tryptophan biosynthesis. Normally, TRPB functions only in the presence of its partner, TRPA, shown here in red. TRPA produces indole, which TRPB takes and couples to serine to generate tryptophan. The ultimate goal of this project is to evolve TRPBs capable of taking derivatives of indole and synthesizing the corresponding unnatural tryptophans, which are valuable chiral building blocks in chemical synthesis. This means we want to evolve TRPB to do three things. One, we want it to act as a standalone enzyme that doesn't require TRPA, so that we can just feed unnatural indoles to the enzyme rather than relying on TRPA. Second, we are interested in having TRPB, which is a thermophilic enzyme, operate at lower temperatures that are more compatible with unstable indole analogs and tryptophan products. And of course, third, we want TRPB to accept many different unnatural substrates to make many different tryptophan analogs. To address these challenges, we encoded TRPB on OrthoRep and deleted TRP5, which is the yeast homolog of the TRPA-TRPB complex. We then simply grew these cells in diminishing concentrations of tryptophan in the presence of indole, thereby challenging TRPB to couple the indole to serine in order to provide the cell with its essential tryptophan. This resulted in significant adaptation, as shown by these outcomes on the right of the slide, where we are getting multi-mutation TRPBs to do the job after about 200 generations of culturing. The evolved TRPBs are indeed quite active. When we encoded them in a fresh strain to test their activities, we found that the evolved TRPBs could support full growth of the cells, matching wild-type growth rates represented by the gray TRP5 bars. This occurred only in the presence of indole, suggesting that evolved TRPBs were operating as desired. This high activity held up in vitro studies. Here, the Arnold group expressed our variants and found that in a test tube, they could produce tryptophan from indole and serine with higher productivity than a previously engineered benchmark version of TRPB called TM triple. What we were also glad to find was that the increased activity of evolved TRPBs transferred to other indole substrates as promiscuous activity, producing unnatural tryptophans, which was our ultimate goal. At the beginning of the project, we were planning to do further evolution specifically for using unnatural indole analogs, by using biosensors for unnatural tryptophan as our selection. But because the promiscuous activity here was high, we decided to take a different approach to access new unnatural tryptophans. Specifically, we just ran more replicates of our TRPB evolution experiment, which is, of course, easy to do, since it just involves passaging cells in diminishing concentrations of tryptophan. The hypothesis, which proved to be correct, was that independent evolution experiments would result in a diverse array of highly active TRPB variants, whose diversity would cover other substrates that we didn't explicitly challenge TRPBs to use. What you see in this heat map are several TRPB variants taken from 10 independent evolution experiments and their in vitro performance in synthesizing tryptophan or tryptophan analogs from a variety of indole derivatives. As you see, again compared to the benchmark TM triple, our evolved variants perform quite well. And across the panel, all of the indole derivatives we tested had at least one TRPB variant that could use it to make the corresponding tryptophan analog. I want to just highlight some of the data from the heat map. On the left here, you see that there are several TRPBs, F7, D2, and F1, that have high activity for using a broad collection of indole substrates. In the middle, you see that many of the evolved TRPBs are selective for 7-bromoindole over 5-bromoindole, which may be useful in cases where we want a specific tryptophan analog produced from a messy mixture of indole substrates. Finally, on the right, you see that there is a variant, G6, that has activity with 5-trifluoromethyl indole, which is a substrate that no previous engineered or evolved TRPB variant has been able to make use of and make the corresponding trifluoromethyl tryptophan from. So the immediate significance of this study is that we now have a new set of TRPBs that can be used for the synthesis of a broad range of tryptophan analogs. This will be useful commercially. The larger significance, though, I think, is that OrthoRep allowed us to run many independent evolution experiments, each resulting in significant adaptation towards high enzyme activity on not just the evolved activity, but also on related activities. This is sort of like nature, which runs evolution at such a scale that it finds not just functions under selection, but also related functions. Now that OrthoRep can take the same approach, but on laboratory timescales, it should be useful in many other enzyme evolution projects to come. I want to conclude by telling you why I'm excited about OrthoRep and what the future for OrthoRep and similar systems hold. So, OrthoRep belongs to an emerging area of research aimed at achieving continuous evolution, where we can evolve new gene and protein functions rapidly in an essentially autonomous manner, for instance by just passaging cells. The reason this research area excites me, and the many others on this list who are working also in continuous evolution, is because continuous evolution, I will argue, achieves depth and scale in evolutionary search to great effect. Let me tell you what this means by contrasting continuous evolution with classical approaches to directed evolution of proteins and enzymes. Traditionally, if you want to evolve a new function from a gene, you run the process shown here, where you take a gene of interest, mutate it in a test tube to create a library of variants, clone that library into cells where the variants express and confer some selectable function, select for your desired function, isolate the winners, and repeat. You'll note that this process can be divided up into some in vitro steps and some in vivo steps, which makes sense under the error threshold discussion I began my talk with. To achieve protein evolution, you need very high mutation rates to sample diversity. These high mutation rates are not accessible inside cells, so you need to do them in a test tube. But then, you need to be inside the cell to express genes and carry out functions that you select for. So you end up doing this cycling between in vitro and in vivo steps. This cycling, however, requires you to manually stage the evolution process, which is a highly onerous task. As a result, you can't easily achieve depth in evolutionary search because each cycle is only a step in adaptation, and running cycles over and over again becomes quite burdensome and time-consuming. You also can't easily achieve scale in evolutionary search, since each independent experiment would compound the burden of this manual staging. What continuous evolution does is it replaces the stepwise manually staged evolution process with a much more natural one, simply involving culturing cells over time in the case of OrthoRep. And since culturing cells can be done for extended periods of time as well as in many replicates, you can achieve depth and scale in evolutionary search. My excitement for continuous evolution in general, and OrthoRep in particular, comes from the potential power of depth and scale in evolutionary search. First, depth. Depth is important if you want to access ambitious functions, because ambitious functions are probably many mutations away from where you start. Perhaps high activity of the s.p.bs we evolved are an example of this. Depth is also important if you want to use different selection schedules to cross fitness valleys. In evolution, one of the key questions is how do you go from one fitness peak to another fitness peak that is higher? In other words, how do you avoid getting stuck at a suboptimal fitness peak? A very important way to become unstuck is to fluctuate your selection, for instance by imposing some period of drift where you relax selection to allow your population stuck at a peak to spread out, crossing fitness valleys that may then lead to the next peak. This requires long mutational pathways and therefore only comes if you have depth in evolutionary search. A project I didn't discuss actually shows this. We encoded an enzyme called his-A, which is involved in histidine biosynthesis, and challenged it to evolve activity for a different reaction involved in tryptophan biosynthesis. What we discovered was that his-A would only partially adapt if selected strongly for tryptophan biosynthesis. And if you continued to select strongly, you stayed at partial adaptation. However, If you relaxed the selection and then returned to strong selection for tryptophan biosynthesis, you could adapt fully, producing variants that could support cells to grow at near wild-type rates in the absence of tryptophan. Here I'm just showing some of these variants of His-A which have very high activity in tryptophan biosynthesis, but they are only reliably accessible with some drift. Well, what about scale? In the DHFR experiment, I think I've already shown how scale in replicate experiments can allow us to better map finis landscape. But scale is important for other reasons as well. For instance, if you have a starting point and you have many related activities you want to evolve from it, you need scale. This was demonstrated in the s.p.b case, and I should add also being exploited in work we are doing on antibody evolution that I didn't have time to present. Briefly, in that work, we have encoded collections of antibodies on OrthoRep and can now affinity mature them to different antigens, where OrthoRep is autonomously sampling new diversity to drive the affinity maturation process. A final benefit of scale that I want to mention is what I call strategic generation of evolutionary diversity for protein engineering. We are very excited to use OrthoRep's scalable evolution capabilities in order to make large collections of protein variants that all have evolved a particular function, but in highly diverse ways. The hope is that such diverse data sets will be enormously valuable for feeding machine learning algorithms that can extract fundamental sequence-function relationships behind proteins and subsequently help us design new proteins, an idea we're working on with computational biologists. So I'm looking forward to a lot of new work that we and others will do with OrthoRep and continuous evolution, guided in particular by the opportunities our newfound depth and scale in protein evolution afford. With that, let me thank all the great researchers involved in this work. Arjun, Alex, Ziwei, and Gary are the ones who really built OrthoRep and made it work in the first place. Arjun and Gary led the DHFR evolution experiments. Gordon led the s.p.b evolution work, along with Tina, Ella, and Patrick in the Arnold group. Ziwei led the His-A evolution work that I briefly mentioned at the end. And Alon, Jake, and Jonathan, along with Connor in the Cruz group, are leading our antibody evolution project. Thank you all for listening.